Hello, welcome everybody to this webinar on T cells and COVID-19, translating research findings into clinical options. My name is Kirsten Langefeld. I'm a product manager for ex vivo T cell depletion systems for the productions of cell therapy products. But before we start, please acknowledge the displayed disclaimer. In the first part of today, I will briefly introduce products and workflows that have been optimized to study T cells in SARS-CoV-2 infections. In the second part, I will introduce the product systems of Miltenyi Biotec for the generation of clinical-grade cell products and their use in published cell therapy concepts to fight SARS-CoV-2 infections. Why is it important to study the role of T cells in COVID-19 infections? T cells play a key role in the defense against viral infections, but also in the generation of immunological memory, which will give protection from future infections. Studying T cells is therefore important for research on evaluating immune responses and to investigate options of an adoptive cell transfer for future therapeutic purposes. Let us have a look on tools and technologies to study antigen-specific T cells. So first, T cells need to be isolated from a donor sample. Here, for tissue dissociation, the GentleMACS tissue dissociator is an appropriate choice. Next, the cells are stimulated with the antigen of interest. Please note that antigen presentation cells need to be present to efficiently activate the isolated T cells with a certain antigen. When working with PBMCs, this is already the case. However, with isolated T cells, APCs have to be added separately. Following stimulation, only the antigen-specific T cells will get activated, and the cells can then be stained intra- and extracellularly to assess their phenotype and analyze cytokine expression. Before cell analysis, antigen-reactive cells can also be magnetically pre-enriched using distinct surface markers. And then, finally, the cells can be analyzed using flow cytometry, for example with our MaxQuant flow cytometer, or sorted on the MaxQuant Taito cell sorter. Here, the efficient antigen-specific stimulation of T cells is achieved with our Peptivator peptide pools. Peptivator peptide pools efficiently stimulate both CD4 as well as CD8-positive T cells and recover an extensive portfolio of virus, tumor, fungi, microbiota, and autoantigens. The peptide pools consist of 15 mere peptides with 11 amino acid overlaps, which cover the complete sequence of the respective antigen. Our Peptivators are available in research, premium, and even Max GMP grade, which allows a rapid translation in the clinic. Finally, they are also available in the convenient 96-well format for high throughput applications. So our Peptivators can also help to standardize your T cell assays, as they are ideal simulation reagents for antigens and very specific T cells. Furthermore, I want to point out the cytokine secretion assays of Miltenyi Biotec. These cytokine secretion assays allow the viable enrichment of cytokine secreting cells from mouse and human samples. The principle is rather simple. First, secreted cytokines are immobilized on the cellular surface by the so-called CAT reagent. Basically, thereby, the cell labels itself with its own cytokines. In a second step, the immobilized cytokines are then labeled with fluorophore-conjugated antibodies, called detection reagents. Living cells can then be detected without intracellular staining using flow cytometry. In an optional third step, the fluorophore-labeled cytokine-secreting cells can be specifically enriched and used in downstream assays with the help of anti-fluorophore magnetic beads. Consequently, our cytokine secretion assays come in two versions. For flow analysis only, Miltenyi offers the virus detection kits, while for enrichment and analysis of viable cytokine secreting cells, we have several enrichment and detection kits. For the isolation of antigen and virus-specific T cells in a clinical setting, we offer a clinical-grade cytokine capture system running on our CliniMACS Prodigy platform. For clinical-grade production of cell products against COVID-19 infections, the Peptivator SARS-CoV-2 Select is already available in Max GMP grade. In addition, the CliniMACS system can also be used for the depletion of CD45 array positive T cells to produce cell products that contains memory T cells. So let us now switch to the news from the field on clinical applications. Cell therapy products based on virus-specific donor T cells have been used to treat opportunistic viral infections in the context of allogeneic hematopoietic stem cell transplantation. It has been discussed whether this treatment options could also be applied to COVID-19 patients. In this context, I would like to present the work of 3 different groups from Singapore, the United Kingdom, and Spain. As the authors explain in these publications, the transient presence of donor T cells in COVID patients could be possible under certain conditions. On the other hand, it would be imperative to prevent alloreactive reactions against the patient. Miltenyi Biotec offers workflows for the production of cell products that are used in certain cases to prevent or treat opportunistic infections in patients with allogeneic HSCT. In the publications, the authors describe how these workflows could be used to produce cell therapy products against SARS-CoV-2 infections. First, some background information on T-cell-based therapy against infections. This treatment was originally developed in the context of allogeneic HSCT. Until the reconstitution of their new immune system, these patients are at high risk for opportunistic viral infections as CMV, ADV, EBV, or BKV. Antiviral drug-resistant cases have been treated with adoptive T-cell therapy. Miltenyi Biotec provides tools for manufacturing T-cell-based products that are used to treat certain viral infections in allogeneic HSCT. For 17 years, the clinical application of these treatment options has been continuously published in peer-reviewed journals. By now, new ways to treat COVID-19 patients with adoptive T-cell therapy under certain conditions have been described. Let us start with the concept of transferring virus-specific T cells to combat certain viral diseases. As reported, adoptive transfer of allogeneic VSTs can rapidly restore virus-specific immunity and control viral infections after allogeneic stem cell transplantation. Sources of virus-specific T cells may include stem cell donor-derived VSTs in the setting of allogeneic stem cell transplantation, and third-party donor-derived VSTs, which has also been used in patients with infections after solid organ transplantation. The production of antiviral T cell products requires the enrichment of relevant VSTs and depletion of potentially alloreactive T cells. There are several strategies to produce these products, as for example those that are based on ex vivo expansion. Another option is provided by the CliniMACS Prodigy CCS interferon-gamma system from Miltenyi Biotec. For this option, it is necessary to find a convalescent donor for the desired viral target. How does the CliniMACS cytokine capture system work within the CliniMACS Prodigy process? The CliniMACS Prodigy process integrates all critical steps of the CCS workflow, from antigen-specific T cell stimulation to labeling of target cells, magnetic isolation, and formulation within 12 hours of total time. Importantly, all cell processing steps are automated, ensuring a convenient and highly standardized separation process. These include stimulation of the starting product from a convalescent donor with a virus-specific peptide to activate the desired virus-specific T cells, separation of these cells via the interferon-gamma secretion, and the magnetic separation strategy to enrich interferon-gamma producing cells. Recently, we launched a Max GMP Peptivator targeting SARS-CoV-2-specific peptides, and we will be glad to provide the reference list with clinical applications. All necessary consumables for these procedures are supplied by Miltenyi Biotec. The CliniMACS Prodigy CCS interferon-gamma system is available as a CE-marked medical device application. Outside the EU, regional legislation and regulations are applied. The clinical use of the cell product is subject to regional legislation and is under the responsibility of the user. Let us now take a look at the second possibility of producing cells against certain infections. Donor-derived CD45 array depleted cell products have been used in the field of allogeneic HSCT. Memory T cells provide immunity against many viral infections that the donor has experienced during his lifetime. Prior to clinical application, it was shown in mouse models that the alloreactive potential is high in the naive T cell compartment and significantly reduced in the memory T cell compartment. CliniMACS CD45 array depleted cell products are used to treat opportunistic infections and to enhance immune reconstitution in the setting of allogeneic HSCT. CD45 array is an isoform of the CD45 family that is present on 100% of naive T cells, but not on CD45RO positive central and effector memory T cells. It is also present on approximately 100% of B cells, 80% of NK cells, and 30% of monocytes. CD45 array is not a typical T cell marker, but it is an ideal marker to deplete alloreactivity from a product in one step, leaving specific memory T cells with the given benefits, and thus represents an excellent production option for clinical applications. Miltenyi Biotec offers two technical options for the clinical grade depletion of CD45 array positive cells from leukapheresis products, performed on either the CliniMACS Plus device or the CliniMACS Prodigy device. The starting fraction is labeled with antibodies targeting the surface marker CD45 array. Magnetic separation results in a target cell fraction passively enriched for memory T cells. The CliniMACS Plus CD45 array system has already been released to market in 2011. Please contact us if you would like to receive the full list of references for this clinical application. Consumables for these procedures are supplied by Miltenyi Biotec. Two CE-certified medical device applications are available for the depletion of CD45 array positive cells. The CliniMACS Plus CD45 array system provides a semi-automated procedure for starting products, containing up to 50 times 10 to the 9 white blood cells. The CliniMACS Prodigy PP45 array system provides a fully automated process for smaller cell amounts in the starting material. In addition to leukapheresis products, whole blood can also be used as starting material with this system. Outside the EU, regional legislations and regulations are applied. The clinical use of the cell product is subject to regional legislation and is also under the responsibility of the user. Several groups have developed manufacturing processes for cell products from convalescent donors targeting SARS-CoV-2 infections. We present here 3 publications in which these new developments are described. They are all open access papers. Leung and his co-workers describe a process for flexible and rapid manufacturing of virus-specific T cells once a donor is available. This option uses the CliniMACS cytokine capture system, interferon-gamma. The publication from Kuba et al. Describes a process that also starts with the enrichment of virus-specific T cells using the CliniMACS cytokine capture system. The difference to the previous publication is that here a culturing step is added to increase the harvest of virus-specific T cells. The final products can be provided within a biobank. The third publication by researchers from Spain also describes the generation of cell products for biobanking. However, here, memory T cell products are provided that are generated using the CliniMACS Plus CD45 array system. Infusion of these cells in COVID-19 patients has been previously published by Antonio Perez and his collaborators. I would now like to briefly present these important publications. However, due to the large amount of information contained in these publications, I will only briefly touch on selected points. To get a complete picture of these important projects, I recommend reading the original publications, which are all open access publications. The two objectives of the study by Leung and collaborators were to investigate whether sufficient numbers of SARS-CoV-2-specific T cells are present in the blood of convalescent donors. Another important point was to investigate whether it is possible to produce clinical-grade products overnight. Whole blood or leukapheresis products were collected from the convalescent donors, and buffy coats were prepared from the whole blood samples. Leukapheresis products and buffy coats were stimulated with overlapping peptides of SARS-CoV-2-specific S, N, and M proteins. The stimulated products were processed by CliniMACS cytokine capture interferon-gamma procedures, and the resulting fractions were analyzed for the amount of interferon-gamma producing cells, their phenotype, T-cell receptor V beta specificity, and functionality. The enrichment procedures resulted in final products that contain a median of almost 1 times 10 to the 6 interferon gamma positive T cells. To demonstrate functionality, one of the samples was successfully re-stimulated with peptide-loaded dendritic cells. Subgroup analysis revealed a relative depletion of naive T cells and an enrichment of effector memory T cells in the enriched fraction, suggesting that alloreactivity was reduced. T-cell receptor V beta spectral typing showed markedly high oligoclonal peaks in the final products. They also concluded that overnight production of clinical-grade SARS-CoV-2-specific T cells is feasible for therapeutic purposes. The alloreactive potential was reduced while virus-specific T cells were retained in the product. They also suggested a dosage of 5 times 10 to the 3 SARS-CoV-2 T cells per kilogram based on previously administered cell numbers in other clinical settings. Under this assumption, 1 unit of whole blood could be sufficient to produce 3 to 6 products for adult recipients. The authors note that, based on previous studies by other authors, virus-specific T cells from donors who shared only one HLA molecule are effective. Based on this information, for the 6 donors in the study, the probability of potential patients having at least one HLA match would be more than 88% for Caucasians, more than 95% for Chinese, more than 97% for Malays, and more than 99% for Indians. All these and much more details can be found in the original publication as given below. The group of John Campbell and his collaborators is featured in the forthcoming publication by Rachel Cooper et al. The goal of this study was to characterize SARS-CoV-2 peptide-specific memory T cell populations in convalescent donors. In addition, the authors aim to investigate the feasibility of isolating and expanding these T cells on a clinical scale using an integrated cell culture step. The experimental design included scale-up experiments, functional testing of the obtained cells, and experiments comparing blood from convalescent donors with blood from donors who have not undergone SARS-CoV-2 infection. Important findings of this study are that generation of interferon gamma responses in SARS-CoV-2 virus-specific T cells from convalescent donors is possible after peptide stimulation, isolation, and expansion. Another important finding was that the presence of HLA class 1 and 2 alleles is widespread in the UK population, and that some specific HLA subtypes with strong peptide presentation ability were identified. Finally, the authors concluded that their method for generating large amount of virus-specific T cell products is feasible for clinical trials, supporting severe SARS-CoV-2 infections with comprised endogenous T cell responses. Here we can get an idea of the production method developed by John Campbell and his collaborators in Edinburgh. To begin with, a leukapheresis product is obtained from a convalescent donor. The VSTs contained therein are stimulated and enriched with the CliniMACS Prodigy CCS interferon gamma system and appropriate SARS-CoV-2 specific peptides. The harvested SARS-CoV-2 specific T cells are then cultured for 21 days with irradiated cells from the non-target cell fraction as feeder cells. As indicated, the final cell product contains between 10 to the 10 and 10 to the 11 cells. Restimulation experiments with dendritic cells demonstrated virus-specific T cell functionality after cultivation. The final product is then frozen and can be stored in a biobank system. After 14 days of expansion, central memory CD4 positive cells are the predominant subtype. Furthermore, and as stated, the expanded virus-specific T cells showed negligible co-expression of T cell exhaustion markers PD1 and TIM3 in both the CD4 and CD8 compartment, indicating the cell culture expansion has not induced an exhausted T cell phenotype. All this and much more details can be found in the original publication, and you find the information below. Antonio Paris and his collaborators from Spain have developed a different method for delivering cell therapy products to COVID-19 patients. They use CliniMACS CD45 array depletion to generate products that are stored within a COVID-19 biobank from convalescent donors. An important question was whether SARS-CoV-2-specific memory T cells exist in convalescent donors and whether they can be enriched by CD45 array depletion. This was shown in experiments with PBMCs from uninfected and convalescent donors. In stimulation experiments with three peptides, S, N, and M, interferon-gamma secreting CD45 RO positive memory T cells were detected in convalescent donors but not in uninfected donors. A clinical scale product was generated using the CliniMACS Plus CD45 array system. Again, interferon-gamma secreting CD45 RO positive memory T cells could be generated in the target cell fraction. To check functionality after freezing and thawing, the final product was frozen, then thawed and stimulated with EL-15 for 72 hours. And the subsequent phenotype assay revealed a phenotype characteristic of an activated stage. Here we can get an overview of the production process. A leukapheresis product was obtained from a convalescent donor. As reported, after a CliniMACS Plus CD45 array depletion procedure, 80% of the cells were CD45 array negative T cells. About 84% of the T cells were CD4 positive, and about 14% were CD8 positive. After simulation with SARS-CoV-2-specific peptides, the percentage of interferon-gamma secreting cells in the T cell subsets ranged from 0.31 to 0.38%. The predominant phenotype was central memory T cells. And if you would like to see the entire publication, please go down to the information and have a look at the entire publication. Recently, the use of CD45 array depleted memory T cells in the treatment of COVID-19 patients has been reported by Antonio Paris and his co-workers within a phase 1 clinical trial. Eligible participants were hospitalized patients with PCR-confirmed SARS-CoV-2 infection and, according to further inclusion criteria regarding pneumonia, lymphopenia, and oxygen demand. Enrollment was based on match with the HLA genotype of the convalescent donor and according to inclusion-exclusion criteria. Single infusions were applied in a dose-escalating manner. 10 patients were enrolled and 9 patients were treated. 3 patients each received 3 escalating doses ranging from 1 × 10⁵ cells per kilogram to 1 × 10⁶ cells per kilogram of CD45 array negative memory T cells. The clinical outcome of the study is described in the publication cited below. The author stated that this study provided preliminary evidence supporting the idea that treatment of COVID-19 patients with moderate to severe symptoms using convalescent CD45 array negative memory T cells was feasible and safe. This clinical application is currently being further investigated in the phase 2 clinical trial. All this and much more details can also be found in the original publication as given below. With that, I end today's webinar and thank you for your attention. If you have any questions, please ask them directly or write them to us later in an email.